Hello students, starting with the chapter Protoplasm. Today we'll study next biomolecule nucleic acid. Nucleic acids. They are hereditary material of living organism. They are mainly of two types. They are of two types. First I will going to discuss is DNA. DNA is deoxyribonucleic acid. Next, DNA is macromolecule made up of two anti-parallel chains held together by hydrogen bond. Next is DNA has diameter of 20 angstrom. One turn of spiral has a distance of 34 angstrom, and distance between two adjacent nucleotide is 3.4 angstrom, as there are 10 base pair in a turn. Next, this DNA is made up of nucleotides. So we'll discuss what are nucleotides. Nucleotides, they consist of two parts, nucleoside and phosphoric acid, which is having H3PO4 group. Next, nucleoside is made up of two components. This is, first is pentose sugar and next is nitrogenous base. Pentose sugar is 5 carbon containing sugar. There's a different. Ribose sugar is present in RNA. Deoxyribose is found in DNA. Nitrogenous bases is further divided into two parts, two types. First is purines. Purines are heterocyclic compound. And next is pyrimidines. Pyrimidines are single cyclic compound. Purine is of two types, adenine denoted by A and guanine denoted by G. Pyrimidines is further of three, thymine denoted by T, cytosine denoted by C, and last uracil denoted by U. These four, A, G, T and C, are present in DNA, while this is the nitrogenous base which is only found in RNA. This is the nucleotide and the DNA is made up of these nucleotide units. Now we'll see the double helical structure of DNA. Structure of DNA. Double helical structure of DNA is made up of two polypeptide chains interweaving each other. Here, as we have discussed, the diameter is 20 angstrom, while one base pair, one helix having a distance of 34 angstrom, which is having 10 base pair. So there are 10 bases in the one turn with a distance of 3.4 angstrom in between two bases. Here, this is the double helical structure of DNA, which is right-handed fork. The backbone of DNA is made up of phosphate and sugar moiety. They are alternately arranged in a polypeptide chain. One phosphate is joined by other sugar moiety and forms a DNA backbone. In the helix of DNA are anti-parallel. Anti-parallel signifies one strand will move from 5' to 3' while other will move from 3' to 5'. Here, this is a single helix of phosphate sugar. Similarly, the other nucleotide chain forms with phosphate and sugar moiety. Phosphate, sugar, again phosphate, again sugar moiety, sugar, then phosphate, then sugar moiety, then form the phosphate, then again form the sugars, then again form the phosphate. These strands, as I told you, is anti-parallel. So one will move from 5' to 3' direction and other will move from 3' to 5' direction. The phosphate and sulfur backbone will now attach the nucleotide bases, which are adenine will always pair with thymine of the other sugar in next polynucleotide chain, which is further added joined by a hydrogen bond. There are two hydrogen bonds between AT and there are other guanine will attach to cytosine having 3 hydrogen bonds. So AT, adenine and thymine, one purine will attach to other pyrimidine. AT having 2 hydrogen bond while GC will have the 3 hydrogen bond. Similarly and vice versa, T will pair with adenine with 2 hydrogen bond. Similarly, cytosine will pair with guanine with 3 hydrogen bond. So in this manner, the DNA structure is interwoven 2 helix. This is the structure of DNA. Adenine will join with thymine with 2 hydrogen bond. This can be shown A 2 hydrogen bond with T, while guanine will join with cytosine with 3 hydrogen bond, which can be shown G 3 hydrogen bond with C cytosine. Next, both strand of DNA run anti-parallel to each other. That is, one in 3' to 5' and other on 5' to 3'. This is about the DNA. Now we'll discuss the second nucleic acid, which is RNA. Next we'll discuss RNA. RNA is ribonucleic acid. Ribonucleic acid. Structure of RNA is fundamentally same that of DNA, but difference occur. The first difference is thymine is present in DNA and uracil in RNA. Next point, the next difference is deoxyribose sugar in DNA, but ribose sugar present in RNA. Next is DNA is composed of two polynucleotide chain, but RNA consists of only single polynucleotide chain that runs 3' to 5' prime. Now next we're going to discuss types of RNA. There are different types of RNA, but major and types of RNA are of three types. First is ribosomal RNA, second is transfer RNA, and the third is messenger RNA. First we're going to deal with ribosomal RNA, called as rRNA. 80% of total RNA forms the ribosomal RNA. Next, it is the most stable RNA. It is found in ribosome and is formed inside nucleolus. Next, it helps in protein synthesis. As we know, the ribosomes are called as protein machinery. Next is tRNA, called as transfer RNA. It forms 10 to 15% of total RNA, synthesized in nucleus by DNA. Next, it is also called soluble RNA and transfer RNA. Next, it is among the smallest RNA. Next, at the time of protein synthesis, it acts as a carrier of amino acid, and these amino acids will form a polypeptide chain. Next amino acid type of RNA is messenger RNA, called as mRNA. It forms 1 to 5% of total RNA. Next, produced by genetic DNA in nucleus. Next, this process of formation of mRNA from DNA will form an mRNA, is known as transcription. Next, it is also called as template RNA. It forms the base for the synthesis of protein. Next, it acts as a template for protein synthesis. This is about the messenger RNA. Now, the significance of nucleic acid. The major significance is, most important, it acts as genetic material for transfer heredity heredity information from one generation to the next. Next, it acts as a raw material for evolution. Third, it brings new characters or variations in organism. It forms the basis of protein synthesis. Nucleic acid continue the races from generation to generation. So this is all about the nucleic acid and its types DNA and RNA. Thank you.